Hi, in the previous video we were introduced to the category of white blood cell involved in the specific cellular defence, the lymphocytes, and in this one we're going to look specifically at the B lymphocytes. So the B lymphocytes are the type of specialised white blood cell that produce the antibodies against the antigens on the surface of the pathogen, and this is what ultimately leads to their destruction. So, an antibody is a Y shaped protein and they have receptor binding sites which will bind to the antigen on the pathogen. And when this binding takes place, this will inactivate or immobilize the pathogen, halting it in place, preventing it from infecting your body cells or causing any tissue damage. So many different antibodies, because of this complementary shape to the antigen, will bind to many different pathogens and forms the antigen antibody complex. So this causes an accumulation or a build-up of these immobilized pathogens, making it much easier for the phagocytes to then come along and destroy them via the normal process of phagocytosis. So it's a very effective way at dealing with pathogens. However, we know the action of B lymphocytes can go a little bit wrong in some cases, as there may be instances where these B lymphocytes are responding to antigens which are on substances that should be harmless to the body. A good example being pollen and hay fever. So this is a hypersensitive response, almost an overreaction of the immune system, producing antibodies when it's not necessary. So you get this allergic reaction, you get the mast cells releasing histamine and all the signs of the inflammatory process when really there's no pathogen, there's nothing to cause any harm. So let's try a past paper question. Pause the video and have a go at this problem solving question. Hit play when you're ready to reveal the answer. So here we have a table which shows the number of different types of white blood cells found in blood samples taken from a healthy person and from 3 different patients. And you need to work out which of these patients has had an allergic response. So we've got phagocytes, we've got lymphocytes, we've got mast cells, and what we need to do is compare the 3 patients to the healthy person and see what has changed and what changes we would expect during an allergic response. So the correct answer is patient Z. Okay. So the phagocyte numbers haven't really changed, but we do have this increase in both lymphocyte and mast cells, a change that we see associated with allergic responses. So, increase in the B lymphocytes, making the antibodies, and the increase in the mast cells, because we know we've got that association with histamine during allergies. So, hope that was a nice and straightforward explanation of B lymphocytes, but if you do have any questions, you can let me know.